Okay, the first thing we're going to cover for this chapter is biofarming. So biofarming is the production of proteins in genetically modified plants and animals. And these purified proteins are actually very useful. So these are useful as biopharmaceutical products. So these are types of recombinant proteins that can be used often to treat disease. And a summary of some of the different types of gene products that have been produced and the conditions that they treat are in this table right here. So an example of this is human growth hormone. So human growth hormone is a hormone that circulates in your blood that can stimulate muscle growth and other growth of other tissues as well. And so there are certain forms of dwarfisms, for example, where the cause of that is the lack of production of human growth hormone. And so you can apply this exogenously. So instead of producing it yourself, if you simply can inject it into the bloodstream, that will do the things that are necessary for that is sufficient for doing the things that are necessary that human growth hormone is necessary for activating growth. And so you can do this. You might think, how am I going to do this? One approach might be just to try to purify from humans. You can get blood samples from humans and then purify it out there. Obviously, that would require a blood source, so you'd have to constantly be getting people to donate blood in order to purify the human growth hormone from there. And so instead, researchers have figured out a way to grow it in cultured mammalian cells. And so these cells have recombinant DNA that induces the cells to produce a large amount of human growth hormone. Another example of this is in treating Ebola. So Ebola virus is obviously a very deadly disease that I'm sure most of you have heard of. A couple of years ago now, there was spread of Ebola in the United States, and approximately 50% of people typically die from the Ebola virus. And so one of the solutions, one of the treatments that you can use are antibodies. So Ebola antibodies are antibodies that target this virus, but typically in a normal disease progression, the disease progression is so fast that your immune system is not able to produce antibodies before the Ebola virus is able to kill you. So if you knew as soon as you had Ebola, if you had a way to inject these antibodies into your bloodstream to fight it off, then that would be a possible treatment. And so that's the idea behind that. So you can produce Ebola antibodies by using transgenic tobacco plant. So this is a plant that's able to actually produce the Ebola antibodies. You purify it from that, and then you can just use it as a pharmaceutical. And so one thing you'll notice is that there's a whole number of different conditions that are treated that way. There's a whole number of different proteins that you use to treat it, and these are grown in a whole bunch of different types of organisms. So you have bacteria cells, you have mammalian cells, and then you also have live animals. So chickens, sheep, pigs, rabbits, goats, plants, so bananas, soybeans, tobacco, and then yeast as well. So yeast is just a single cell organism. And so why the complexity? Why do you have so many different approaches? And that is typically due to the complexity of the protein. So let's go over an example. So one of the first success stories of this is using insulin to cure diabetes, or not cure, but at least treat diabetes. And so diabetes is a disease caused by the lack of the insulin protein. And the cause of the disease is the fact that your cells are not appropriately taking up glucose from the blood. So you eat a meal, and that meal is processed into things like proteins, and those proteins are broken down into amino acids, and then the sugars are also taken up by your intestine. And the sugars eventually are going to be in your bloodstream, and if the glucose is circulating in the blood, it needs to be actively taken up by cells that it comes into contact with. So muscle cells that are in contact with the blood, those have to actively transport glucose from the blood. It's circulating in the blood, and you have a cell right here, that cell has to actively bind to it and bring it into the cell. And if it doesn't do that, then the glucose is just going to continue to circulate through the blood. And so you need to have a trigger. And so this trigger that causes the cells to take up glucose is this hormone called insulin. So insulin is released by the pancreas, and this insulin also binds to the cell, and then that activates the pathway to bring glucose into the cell. And so diabetes is a condition where your cells are not appropriately taking up glucose from the bloodstream. And this actually leads to toxicity. So the toxicity of the excess glucose in the bloodstream leads to damage of your eyes, kidneys, and nerves. It causes heart disease, stroke, and eventually you often have to amputate limbs as well. And essentially it's caused by a breakdown in insulin signaling. So there are two ways that this breakdown occurs. So one breakdown is when insulin isn't produced. So if your pancreas, the cells in the pancreas don't produce insulin, they don't release it into the blood, then you never have insulin to bind to the cells. The other defect is when receptors ignore the insulin. So you have the insulin in the blood, but for some reason the cells are no longer activated by it. So they're no longer binding it, or if they're binding it, they're no longer actually changing and operating the intercellular signaling pathways that causes it to take up the glucose. So type 1 diabetes, you might have heard of type 1 and type 2 diabetes. So the first one is when insulin isn't produced, and the second one is when receptors ignore insulin. And so you can ameliorate this disease, you can help this disease by injecting artificial insulin into the blood. And so here we see an individual that is testing their glucose levels. So you simply prick your finger in order to get a blood drop, and then this machine right here can test the levels of glucose. And so if you have too high of glucose levels, then you need to inject insulin, have some sort of injection of insulin, so that your body, so you can force your body to take the glucose up out of the bloodstream. And so again, you have to have some sort of source of this. You could potentially purify it from human blood, but it'd be much easier if you could biofarm this, grow this in a particular organism. And this is no longer how insulin is produced, but at one time it was, and recombinant engineering was used to trick bacteria or to force bacteria to grow something that could eventually become insulin that we could put in this bottle. And so the details of this are a bit complicated, and the reason the details of this is complicated is because proteins are often kind of complicated. So an active insulin molecule we see down here, this is actually a dimer between two different polypeptides. So there's two polypeptides, and these are covalently bonded together by what's called disulfide bonds. And so normally what happens is you have your gene is expressed into mRNA, the mRNA becomes this polypeptide chain, and then this polypeptide chain is cleaved and then reformed together to form this disulfide bond. And so in order to have active insulin, all of these things must happen. You must have something that looks like this. And so if you just express this gene in E. coli doesn't have many of the accessory factors that are necessary for, and this is actually incorrect, I need to remove this from the, sorry, this is not encoded by two genes, that was a mistake. The E. coli bacteria is unable to do this processing. So often proteins need some sort of additional processing in order to make them active. And so the E. coli bacteria is not able to cleave these. And so the solution to this problem was to, instead of expressing just this entire gene of insulin, instead you break it up into these two fragments. And so you have one plasmid that has part of the gene, and then you have another plasmid that has another part of the gene. And so this is a recombinant DNA, where you've taken human DNA and combined it to bacterial plasmid. And of course, this bacterial plasmid has things like antibiotic resistance, it has a promoter region, and then it also has the LAC-Z gene. And so what you're essentially producing is actually a fusion protein between the insulin subunit and LAC-Z. So you transform E. coli, you select for bacteria that are resistant to the antibiotic, and then you induce them to produce a large amount of this fusion protein. So you grow a large number of cells in a bioreactor, and you wait until they've produced a large amount of protein, and then you extract and purify this beta-gal insulin subunit fusion protein from each of these cells. Then you treat the purified protein with cyanogen bromide, and what this does is it cuts at methionine amino acids, and so it's a way to cleave off the LAC-Z from the two parts of the two insulin subunits. And then you simply mix these together, and you induce them to form the disulfide bonds, and so now you have functional insulin that you can add into the tube, and then people can take this at home. So the process hopefully somewhat makes sense in that you can see how you use recombinant DNA in order to express a protein that has therapeutic properties. But you'll also notice that it took a lot of knowledge to do this. So this is why fundamental research is so important, is that we need to have that knowledge to build upon. We need to know how insulin works. We need to know the molecular details of how insulin works. We need to know that we have this cyanogen bromide that can cut protein at specific places. And by building up this knowledge base, we're able to actually increasingly potentiate the types of things that we can build. Another example that we'll go over is the production of antibodies to fight Ebola. So antibodies, so these are proteins in the blood that recognize specific molecules, and these are very important parts of the innate immune system. Your acquired immune system. So this is also a very important part of vaccines. So as we're designing vaccines for COVID-19, essentially what we're doing is we're trying to trick the body into producing antibodies that would recognize the COVID-19 virus. And so the molecular details, again, this is a result of lots of study of acquired immunity, lots of molecular studies. And so we have a pretty good detailed understanding of what an antibody looks like. And so this Y structure right here represents an antibody, and you'll notice it's made of multiple peptide chains. So we have a heavy chain and a heavy chain. We have a light chain and a light chain. So these are 4 peptides. And the reason they're all connected is that there's these disulfide bonds. So this is the second time we've seen disulfide bonds. So these are just sulfur molecules that covalently bond together by a specific type of chemical reaction. And so now these polypeptide chains are joined together here. So the basis of this is that your body produces a large number of these antibodies, and each of these antibodies have very variable regions at their tips. And these are the parts of the antibody that are designed to recognize things. So they might recognize bacteria, they might recognize a protein, they might recognize a virus. And you combinatorially express a large number of these, so your potential to recognize things is very high. And what they recognize is called an antigen. And so this antigen for this is complementary to the specific sites. So what happens if you have an infection, you have a bacteria? Some of these antibodies will be able to recognize and bind to this bacteria. And if these antibodies recognize and bind to it, they can recruit, for example, special cells that are able to eat what it binds to and destroy what it binds to. But the cells can also recognize when this happens, when the antibody has some sort of useful feature. And so this antibody that recognizes Ebola, that recognizes COVID-19, that recognizes any sort of bacteria, they can actually be boosted within the body. And so the the cells that are actually making these antibodies can be recognized and then expanded so that you produce many, many, many antibodies. And so your whole blood is kind of infused with these things that can recognize wherever the infection occurs. So what we want to do is we want to produce antibodies that can fight Ebola virus. And so if we get an infection, then we can simply already have these antibodies made, and then they inject it into humans, and then they recruit the cells, and the virus is eaten, and the infection is fought off. And so this was accomplished by using mice to actually produce these antibodies. So we inject mice with Ebola virus, and then the mice makes many antibodies, and some of these antibodies recognize and block the viral infection. So now we want to recognize the ones that are affected this way. So again, you might be thinking, okay, if we want to do this, why don't we just get like a huge mouse colony of 1 million mice? We inject Ebola into those mice, and then we collect their blood, and we have antibodies from that. So that obviously is a huge scale of death that typically we're not comfortable with in this country, although not completely uncomfortable with this, unfortunately, in this country. But we also have issues where each mouse will produce a different level of antibody blocking. So some of those mice will be much better at producing antibodies that block the virus. And so we don't want to have that uncertainty. We don't want to have that the possibility that we create some sort of antibody that doesn't work very well. And so what we want to do is identify the mice that have effective antibodies, and then identify the cells that actually produce those antibodies. And so there's an approach called plasma cells. So there's an approach to identify these, and we can identify the cells that are producing those antibodies that are very effective. And within those cells, they encode that specific antibody version that can do that so effectively. And so this is actually a violation, just as an aside, this is a violation of how you normally think of mitosis, where we produce genetically identical copies. The immune system is an important exception, where the genetic differences are important for producing different types of antibodies. So we isolate those genes that encode the antibodies, and then we introduce those into plants. And now we can grow and extract antibodies from plants. And so everything that we produce from these will have the exact same antibody. And so now we have repeatability, where each bottle that we produce should have the exact same effect at blocking Ebola. So in this case, why do we use plants? And again, it comes down to the fact that specific proteins are modified, and each protein is kind of modified in their own unique way very often. I mean, there's commonalities. We've seen these disulfide bonds happen, multiple times. But other things like carbohydrates, phosphorylation, those things also happen to cells, ubiquination. So there's all sorts of post-translational modifications that can occur. And so bacteria, because they're so different from humans, they don't actually have many of those factors that allow that. And so this is why we have developed so many different eukaryotic hosts in order to find a host that is most effective at growing whatever protein we want. And then the final thing that we'll cover for this is using recombinant DNA approaches to create vaccines. And so this is kind of like the previous. Before, we were trying to create purified antibody proteins, but now we just want to approach in order to trick our immune system into producing antibodies for a given virus or bacteria. And so, the idea behind this is that you do it before you're infected. So the idea in the last section is that you get infected with Ebola, and then you inject the antibodies. For producing a vaccine, the idea is to inject this before you get it, and that prevents you from ever getting it. So COVID-19 is obviously, this is a huge area of active research in order to rapidly generate vaccines that we can all use in order to build immunity. And so how do you do that? So you trick your immune system to recognize a live virus. And so this is a live virus, but obviously you don't want to inject a live virus into your blood, because then you'll just have an infection. And so instead you want to modify it in some way so that it still causes your immune system to recognize it, but it no longer has that infection potential. And so how do you do that? So a very straightforward way is that you just inactivate the virus. So you take whole virus particles and maybe you bathe them in UV light. And so by bathing them in UV light, you damage all of the DNA and RNA within the virus so that it's unable to use that as a copy in order to replicate. And so this is essentially a dead virus, but it still has the shell. It still has everything on the exterior. You also might attenuate the virus, where you weaken the virus, so it's still alive, but it's not strong enough in order to overcome your immune system to cause an infection. You can synthesize, so you can artificially synthesize virus-like particles. So MG Finn here at Georgia Tech is one of the pioneers in this type of approach. You can also use recombinant DNA in order to produce protein from the virus. And so the virus has a large number of proteins within it, but some of these are exposed to the outside of the viral particle, and probably would be what the antibodies would recognize anyway. So maybe the antibodies primarily try to recognize this protein in blue. And so instead of actually trying to inject something that looks like a virus, what if we can get away with just injecting a purified protein or a synthetic peptide or just some sort of part of it that would go in your bloodstream, and then your antibodies could learn to recognize like that set of sequences. So you're not injecting the viral particle, all you're injecting is protein. Or what if you could just inject DNA that your body can use to produce into this protein, and then your antibodies, the antibodies that bind it, can be recognized and boosted. And so that's the idea behind this, is that you have bacteria that normally has plasmid DNA. You purify the plasmid, you cut it with a restriction enzyme, and now we add a gene from the virus into that in order to create a recombinant piece of DNA that has both viral DNA and bacterial DNA. And in this case, we use a yeast cell in order to produce the protein. So we grow the yeast in a fermentation tank. And so this plasmid causes the production of a large amount of antigen, the hepatitis B antigen, which would be this protein. And so you can inject this now into your body in order to raise an immune response. And then another type of approach, so this is both RNA and DNA approaches. So some of the vaccination approaches actually kind of are RNA based. Is to, instead of going through this whole process of producing the protein and purifying the protein in order to inject it, is you just inject genetic material from the virus. So if you just inject DNA from the pathogen that you recombine into bacterial plasmid, then potentially you can cause the human cells to produce it, and then that protein can be recognized by the antibodies. Those antibodies can be boosted, and then you now have immunity to that disease. So that just summarizes a bunch of different ways that we use biofarming for biopharmaceuticals. There's a large number of things that we didn't cover, but how the approach works and why this has been such a powerful tool for human health.